Hello, everyone. My name is Alan Hudson. I'm the contact PI for the Immuno-Oncology Translational Network Data Management Resource Center, and we are pleased to be working with NCI to coordinate these talks. With that, I'd like to introduce Dr. Dinah Singer, the Deputy Director of the National Cancer Institute, who will be giving an overview of this seminar series. Dr. Singer.
Thank you, Alan, and I'm glad everyone could join today, and I'm really pleased to welcome you to this inaugural talk in the Cancer Moonshot seminar series. The pandemic has really created a lot of challenges for us, as evidenced by the fact that both Cigall and I are speaking from home rather than to a live audience. But it has had the good effect, I think, of increasing participation in virtual meetings. And it's really in that spirit that the goal of this series is to highlight the accomplishments of the Cancer Moonshot program over the past 3 years. And I thought I would take a minute to put this talk in the context of the Cancer Moonshot, which some of you may not be familiar with. Are my slides up, Alan?
No, they are not. Let us know if you want to launch it from our end.
Okay, sorry, I thought they were up. Are they
up?
Up?
It's coming, yep, it's up.
Okay, sorry about that. All right, so what I'm going to do is start with sort of an introductory few comments to tell you about the Cancer Moonshot, its origins, and what we have been doing with it. So it was established by a presidential memo back in 2016 with 3 goals that were articulated. One was to accelerate progress in our understanding of cancer. The second was to enhance data sharing. And the third was to encourage greater cooperation and collaboration within the scientific cancer research community. So even though the presidential memo established the Moonshot, we didn't actually get funding for it until somewhat later in the year when Congress passed the 21st Century Cures Act. And that act appropriated $1.8 billion over 7 years to NCI to support the Cancer Moonshot initiative. And if you look at the graph, you'll see that the funds were really front-loaded in the first 3 years, 2017 through 2019, to really allow us to rapidly implement, new programs beginning already in FY17. And I'm pleased to say that since the passage of the Act and the appropriation of the funds, we've been working very hard to achieve all 3 goals, and I'll focus first on what we have done to accelerate progress in cancer and in our understanding of cancer. The first challenge that we had in implementing this goal was to identify those areas of cancer research that are really appropriate for acceleration. So not all research can be accelerated by additional support and attention. Some is really still too nascent, and some is too far along. So to identify those areas of research that could be accelerated, a blue ribbon panel was assembled. That panel gave careful thought to what areas should be supported and identified 10 topic areas that are listed here. In addition, they also identified cross-cutting themes that went through all of the recommendations, and those included health disparities and partnerships. Since the Blue Ribbon Panel recommendations came out, NCI has been very busy implementing programs targeted at each of those recommendations, and those are summarized here. The vertical column on the far left lists each of the recommendations, and as you go across the diagram by each fiscal year, you'll see the programs that have been implemented. Now, I don't expect you to read this, but I do want you to appreciate that we've launched over 100 programs since the beginning of the Cancer Moonshot. And during the course of this seminar, starting with today, you're going to hear about many of the accomplishments in those programs. So moving on to the second goal of enhancing data sharing, we take that very seriously. A number of programs sponsored by the Moonshot, in fact, all of the programs sponsored by the Moonshot share their data and their protocols openly through various portals and databases and nodes in NCI's Cancer Research Data Commons. To ensure that the progress being made within the Moonshot is actually widely shared, all publications that have been supported through the Cancer Moonshot must be made publicly available upon publication without any embargo. In addition, any of the underlying primary data that were generated through Cancer Moonshot research projects have to be made immediately and broadly available to the public. Turning to the third goal, encouraging greater cooperation and collaboration. That's another goal that we've taken very seriously and we've actively encouraged. As one example, on the left, last fall, one of the last face-to-face meetings that we were able to have, there was a collaborative meeting that brought together investigators from 9 different Cancer Moonshot networks. And what was really an exciting and highly interactive meeting that led to collaborations across some of those networks. Beyond promoting collaborations within the Moonshot, there are also active outreach programs through direct patient engagement and involvement to bring in the broader community to be aware of the Cancer Moonshot. The seminar series that we're launching today is another example of our effort to reach out to the community with the goal of informing both the research community but also the public about what has been accomplished through the Cancer Moonshot. One of the recommendations that was listed on that earlier slide of the Blue Ribbon panel was to expand our understanding of fusion oncoproteins and their molecular complexes that drive many pediatric cancers. Today's seminar is going to focus on advances being made in understanding one of those fusion events. But before concluding, I do want to thank the NCI staff who really worked tirelessly to try to achieve the goals of the Cancer Moonshot. And I think it's important to point out that in implementing all of those 100 programs that you saw in an earlier slide, no new staff were hired. All of the NCI staff involved with the Moonshot have taken on the challenge in addition to their other full-time responsibilities. And so all of the funds that were appropriated to the Cancer Moonshot have been spent on research. And I especially want to thank the NCI staff who are involved in organizing this seminar series, particularly Lillian Crow and Claire McCarthy and their colleagues. Okay, so now it's my pleasure to introduce the moderator of today's seminar, Dr. Charles Mulligan from St. Jude's, who's also a principal investigator in the FUSE initiative, and he's investigating the role of NUMP98 fusion oncoproteins in pediatric and adolescent leukemias. He's going to introduce our speaker and then moderate the questions after the talk. So thank you very much. And Charles, I'll hand it over to you.
Thank you very much, Dr. Singer. It is my great pleasure to introduce today's speaker, Cigall Kadoch. So Dr. Kadoch is Associate Professor of Pediatric Oncology at the Dana-Farber Cancer Institute, as well as holding faculty positions at Harvard Medical School and being a member and epigenomics program co-director at the Broad Institute of MIT and Harvard. So I think it's true to say that Cigall has had a spectacular career. She, just at this stage, indeed, and she did a PhD with Gerald Crabtree at Stanford, and then established her own lab at the age of 28 at Harvard, and since that time has established herself as a leading light in the field of chromatin and gene regulation, which is critical to many areas of cancer, including the function of fusion oncoproteins. Her lab studies the structure and function of chromatin remodeling complexes, particularly the SWISNF or BAF complex, which has gained increasing importance, particularly from genomic studies that have shown it to be one of the most frequently mutated targets in cancer writ large. Her groundbreaking work has been recognised by numerous awards and recognitions, including an NIH New Innovator Award. She's a Pew Scholar, an ACS Research Scholar, and recently was named the American Association for Advancement of Science Martin Rose Wachtel Cancer Research Prize recipient. So, Cigall, it's our pleasure to hear your talk, and over to you.
All right, I'm just sharing my screen. One moment. Let's try this. Share screen. There we are. Wait a second. Let's try this then. Can you see my screen?
Yes, yes, it's up.
Fantastic. All right. Well, thank you so much for that incredibly kind introduction, Charles. And I really want to thank and extend my appreciation to Cancer Moonshot, to Dr. Singer, Dr. Woodkin. I'm really very honored to give this inaugural talk for Cancer Moonshot. So as some of you may know, our laboratory is interested in understanding the structure and function of large ATP-dependent chromatin remodeling complexes, most notably the mammalian SWISNF, also called BAF, family of chromatin remodeling complexes. And we place particular emphasis on understanding how perturbation to the individual components of these regulators can play driving or contributing roles in human disease. And in this talk, as Charles and Dina mentioned, I'll focus largely on our efforts that have been supported by the U54 that we hold for fusion oncoproteins within the context of Cancer Moonshot. So our laboratory is largely a basic science laboratory focused and centered in biochemistry, biophysics, increasingly structural biology, genomics, epigenomics, and even systems biology. We really take a combination of approaches to understand the many angles by which this chromatin remodeling complex and others can be perturbed in human disease. And in parallel, I'm fortunate to partner with and participate in groups intensely dedicated toward translating such discoveries into hopefully meaningful therapeutics for patients, which brings me to this disclosure slide. The scientific founder, BOD member, SAB member, shareholder, and consultant for Foghorn Therapeutics, which is a company that I founded several years ago, located in Cambridge, but I will not discuss any off-label use or investigational use in this presentation. So I think this group really needs no introduction, but of course the sequencing of the human genome and subsequently disease genomes has immeasurably advanced our understanding of the genetic underpinnings of human disease. Particularly the TCGA Cancer Genome Atlas effort has heralded a number of exciting discoveries regarding the genetic basis for numerous cancer types. I'm consistently inspired by this effort, and it really continues to serve as the motivation and inspiration behind much of our work. In this group, as many are familiar, sequenced a number of different cancer types, tens of thousands of tumor samples, identified over 300 driver genes and over 3,000 putative missense driver mutations. So really that effort, I think, continues to serve as inspiration, not only for our group, but for many around the world, and certainly in the field of chromatin regulation. So this is actually one of my favorite panels from the Bailey et al. Cell paper in 2018, which detailed the collation really of all of these TCGA efforts. And what we can see here with the various tumor types around on the x-axis, a number of different pathways that are perturbed with the various frequencies shown here by fraction of genes. You can see a number of culprits that have been longstanding drug discovery culprits for a number of years now. So signaling pathways, TGF-beta signaling, RTK signaling. We see other common culprits, apoptotic regulators, cell cycle regulators frequently mutated in disease. But perhaps the most unexpected and I think exciting element of this sequencing effort was really the previously unappreciated notion that chromatin regulatory proteins, or genes rather, are mutated very frequently in human cancer. So we see, chromatin modifiers, histone modifiers, DNA modifiers, genes encoding members that can play histone modifications. And the complex I'm going to tell you about today is so frequently mutated it gets its own row. So we see very frequent mutations across a number of cancer types, as well as mutations in transcription factors, which of course leverage the various facets of the chromatin landscape to elicit their cell tissue, and ultimately disease-specific phenotypes. So as I mentioned, these mutations we see are in genes encoding proteins and protein complexes that render chromatin in the closed state, for example, PRC2 complexes and polycomb repressive complexes. We also see genes that are mutated that encode proteins and protein complexes that help open chromatin and enable active DNA accessibility and active transcription. Some of these include DNA modifiers, histone modifiers, histones themselves, histone tails, and importantly for the subject of this talk, ATP-dependent chromatin remodeling complexes. This is an artistic schematic of the ATP-dependent chromatin remodeling complex most studied by our group, Mammalian SWI complex, drawn approximately to scale here with the local nucleosome environment. These complexes were originally discovered in yeast in screens for mate type switching and sucrose fermentation. They were then later characterized in Drosophila and ultimately in the human cell context. They utilize the energy of ATP hydrolysis to essentially mobilize nucleosomes, thereby enabling timely and appropriate accessibility for transcription factors and transcriptional machinery to elicit timely and appropriate gene expression. Now, for this particular family of chromatin remodeling complexes, they are comprised of the products of 29 different genes shown here on the left. And these 29 genes assemble to create for 3 distinct, this is shown just as one for simplicity, but 3 distinct final form complexes, wherein each individual complex has hundreds of possibilities because multiple positions within the complex can accommodate one or another paralog that sometimes can be co-expressed in a given cell type. So that's true, for example, for SMARCB1 and SMARCB2 or BRG1 BRM. These are the 2 ATPase paralogs in the complex. That's also true for ARID1A, ARID1B paralogs within the complex, and so on and so forth. So with all this combinatorial complexity, what makes this challenging to study is that there's actually hundreds of possible combinations in any one given cell binding chromatin that we have to work with here in trying to identify which type of complex is most at play in any one given disease entity that we study. So in examining the TCGA data, we can see these are very frequently mutated. We estimate these are in actually over 20% of human cancers. ARID1A is the most frequently mutated subunit. You can see here at the top, and mutated in a broad range of human cancer types, whereas other subunits are mutated in a much more punctate manner, as you can see throughout this heat map, in very specific cancer types, one or another. And this doesn't actually incorporate yet the rare cancers that I'll tell you about. If we start to add, in addition to exon mutations, start to add in loss of heterozygosity, deep deletions, this number continues to rise, around 24% of human cancers. So this is really also still the tip of the iceberg because, of course, there are many other ways to perturb genes, whether looking at enhancer structure and other mechanisms that we are actively investigating. Now, in addition to those, what I showed on the previous slide, one of our main motivations for studying this family of ATP-dependent chromatin remodeling complexes has really come from rare cancers. Rare cancers that are often pediatric rare cancers, as shown here on the x-axis, and also why I find my home department in pediatric oncology at the Dana-Farber Cancer Institute. And we can see here that in a number of these very specific types of cancers that are genomically well characterized, mutations in the various components of these chromatin remodeling complexes come near hitting the 100% mark or come close to hitting the 100% mark almost uniformly, explaining various disease types. That is certainly true for synovial sarcoma, which I'll be explaining a lot about today. So some key themes to arrive at after looking at these genomic data. We see that specific subunits can be mutated in very specific malignancies. And actually what I'll tell you about today with synovial sarcoma, the fusion oncoprotein SS18-SSX is only found to be translocated and mutated there in synovial sarcoma, not in any other malignancy to date. Other subunits, as I showed, like ARID1A, have a much more wide range of mutation across many different cancers, so suggesting some common defining commonalities that may be central to a number of different tumors arising from a number of different tissue types. The most frequently mutated subunits, such as ARID1A, for example, are not actually always those required for in vitro remodeling activities. And this is an important idea for us in the field, in the sense that the textbook definition of this complex is that placing this complex on a chromatinized template can result in remodeling of that chromatin template. However, not all of the subunits in their domains are required for that. So this is a really key question, I think, central to the field, and begs the question that we may need additional assays that can more faithfully recapitulate mammalian chromatin. And finally, the mutations can be heterozygous, suggesting gene dosage effects. So over the past 5-6 years, I've had my young lab now. We've essentially walked our way around this complex, if you will, focusing quite a lot on many of the rare cancers where the perturbation in this complex is the driving, often pathognomonic feature for this complex. So that has been true with our work on malignant rhabdoid tumor, epithelioid sarcoma, with these various studies, the colorectal cancer, small cell carcinoma of the ovary, particularly for the ATPase components. We've worked extensively to resolve the biochemical and structural modules in the 2 stories shown here, the functional modules, examining how this complex can interact with transcription factors, which can essentially hijack them to new spots on the genome to guide their activities to those specific loci. And many of these, as shown here, are loss of function contexts in which the mutation that we see is a deletion in a subunit, for example, SMARCB1, biallelically deleted in essentially 100% of malignant rhabdoid tumors, loss of function events or loss of function mutations. One very interesting disease context in which the perturbation of the complex is actually a gain of function within a subunit itself, this is the SS18 subunit, is in synovial sarcoma, which has the SS18-SSX fusion oncoprotein that is known to be driving of that disease. And so we spent a lot of time investigating this particular gain of function entity in hopes that some of the insights and results we see from this type of cancer can also inform a wider swath of cancers that are involving this particular chromatin remodeling complex. So this goes back actually to work I did in Jerry Crabtree's lab as a graduate student, where we first identified SS18 actually as a stable subunit of this complex, and found that in tumors that harbor the SS18-SSX fusion oncoprotein, which results in this characteristic TX18 translocation in synovial sarcoma, that this fusion oncoprotein usurps BAF complexes, displacing the product of the wild type allele, the normal SS18 protein, and seemingly and surprisingly, a subunit called BAF47 or SMARCB1, which until some recent results I'll show you, we didn't really quite understand. But this was the disruption at a biochemical level to the complex, and that biochemical disruption resulted in changes on chromatin and specific gene activation that was really hallmark to synovial sarcoma tumor types. And very excitingly, we found that we could actually reverse this if we simply toggled the concentrations of the wild type form versus the oncogenic fusion oncoprotein form. You could toggle between an oncogenic complex and a wild type complex. So one way that we can achieve that toggling is by using hairpin knockdowns, for example, for the fusion. These are very good knockdown constructs. You can see that this results in a very strong decrease in the 2 bands corresponding to the fusion. And actually this allows for a reappearance and restabilization of the wild type SS18, which is shown here by these 2 bands, and the BAF47 or SMARCB1 tumor suppressor subunit shown here. So we wanted to ask, now that we had a system for doing this, how would the SS18-SSX fusion, by incorporating into the complex, how would this affect the global targeting of these complexes on chromatin, and how would this then impact subsequent gene expression? So to pursue this, at first we used a number of genomic strategies, shown here, ChIP-seq in synovial sarcoma cell lines, where we could appreciate something that was really quite unique for all of the other tumor types that we study relating to this complex in the lab. This was really, this stood out quite a lot to us, and that is that the targeting of BAF complexes in synovial sarcoma, when the complex has this fusion oncoprotein on it, you can see there's a very unique set of sites to which the complex localizes, such that when we remove the fusion oncoprotein by using shRNA, you can see that the complex is essentially released to a number of other sites on chromatin, and these sites tend to be the wild type sites, if you will, to which complexes would normally target in an original cell of origin, for example. We can see that the reverse is also true if we take now fibroblasts and simply introduce either wild type SS18 or this fusion oncoprotein. We can see that the fusion oncoprotein succeeds in dragging BAF complexes to a number of new sites in a de novo manner that it didn't target to before, and that this is accompanied with increased accessibility at those sites as measured by TAC-seq. Now, one other very interesting feature of the synovial sarcoma SS18-SSX fusion oncoprotein is that we noted that it actually targeted BAF complexes to polycomb repressed sites on chromatin. So these are targeting these complexes to sites that should normally be heavily repressed by PRC1 and PRC2 complexes. This is shown here by these immunofluorescent experiments, where we can examine the immunofluorescent signals of Ring1B, the catalytic component of PRC1, and SUSY12, one of the members of PRC2. And you can see here that in the presence and introduction of SS18-SSX, you can see that there's very nice overlap particularly of SS18-SSX with ring 1B, as shown here in these immunofluorescence images, as well as by ChIP-seq. We noted this very interesting pattern whereby the complexes, when the fusion is present, the complexes seem to overlap at loci where the PRC2 complexes are present and their marks are present. You can see again here with these Venn diagrams that really only when the fusion is present by introducing, for example, here the V5 tagged SS18-SSX on the right in fibroblasts, one can appreciate that this is where we see the most overlap. So somehow this fusion protein is dragging complexes to sites it shouldn't be, to sites that are normally polycomb repressed, and then the result of this is gene activation. So our key question, and one of the main questions that inspired our proposal for the FUSE-ONC consortium, was really to understand what is the mechanism that can underlie this very unique site-specific targeting and the unique chromatin interaction properties specifically of this SS18-SSX bound BAF complexes. And the reason we want to know about this is because if we knew this, we would be able to then define the specific features of the interaction of SSX with chromatin, and importantly, to understand the identified cancer-specific synthetic lethal dependencies that we see in synovial sarcoma, and most importantly, to inform and design new therapeutic approaches for this cancer type. So to begin this effort, we started biochemically, as our lab often does, and we simply purified either complexes containing wild-type SS18 or SS18-SSX. We just purified these out of cells, whereby we introduced each one of these in an HA tagged manner, and we purified the complexes from either the nuclear extract, so soluble nuclear extract, or the chromatin fraction. Now, when we do this with wild-type SS18, as we do often with BAF complexes, you can see very nicely that most of the BAF complexes come out here in the soluble nuclear fraction. They're very pure, very clean. You can label all the subunits. However, when we did this with SS18-SSX, you can see the majority of complexes are actually still stuck to chromatin. They're in the chromatin fraction. And very importantly, along with these complexes, almost in a way that was impossible to dissociate, these tether extremely tightly to the histone octamer, to nucleosomes. So much so that when we performed quantitative mass spectrometry studies, we could see that the enriched proteins, upon purifying SS18-SSX1 on the x-axis shown here, we purify a number of members of the complex, particularly the ATPase module of the complex to which SS18 is fused, as well as histones. So you're actually pulling out the histones in almost a stoichiometric proportions here with BAF complex components. And that's something that we've never seen, in any other purification for complexes that we study in the lab. We can visualize this in another way, now performing here density sedimentation studies, examining the sedimentation patterns of complexes now using 10 to 30% glycerol gradients. This is essentially a normal profile of a BAF complex, eluting in fractions 13 and 14, for example, of a gradient. But look what happens when we now have complexes that contain this SS18-SSX fusion. You see that the complexes are really spread throughout the gradient. The majority of the full complex are in a heavier fraction. And you can see that no matter what we do, this is pulling along this histones down here. This histone octamer is being pulled along with SSX wherever SSX is. Another way to examine this is using differential salt studies, where you can really see clearly here that in synovial sarcoma cells that have the fusion, the complexes cannot be eluted even with 1,000 millimolar salt. They're stuck in the pellet. But the second we start to remove the fusion protein by using shRNA, you can see this releases these into the soluble fraction. And this was true as well using FRAP studies, which examines the residency time on chromatin, in that SS18-SSX bound complexes have a significantly longer residency time to recover compared to SS18 wild type complexes. So to examine what are the features of this SS18-SSX fusion that is actually contributing to this, in the previous slides, I showed you all studies examining complexes, BAF complexes that are bound to SS18-SSX. But what about just SSX alone? Could we now try to resolve just the features of the SSX tail that is contributing to this overall very unique set of phenotypes that we see? And indeed, when we purified SSX, we can see that SSX, just the minimal 78 alone, is actually sufficient in and of itself to bind histones. So this is the region that is binding to the nucleosomes. And to get an idea of what types of nucleosomes these were binding to, we simply incubated these with MNase digested mammalian mononucleosomes, which have the constellation of PTMs and other marks on them. These are digested from mammalian chromatin, and then perform quantitative targeted histone mass spec. And what we see here is that SSX alone, just at 78 amino acids, preferentially captures nucleosomes that are decorated with repressive marks. So there's something on this SSX protein that is telling it to go to and prefer nucleosomes that are repressive. You can see here as you add more acetylation, which is generally activating, you see more and more decrease in the ability to bind to those nucleosomes. So to dissect this a little bit further, we performed some amino acid and conservation analyses here, now examining really just the most 34 C-terminal amino acids of the 78 amino acid tail. And we noted by conservation, there was a very conserved basic region shown here and a very C-terminal conserved acidic region. So to try to understand the contributions of these that were quite conserved, we performed mutagenesis throughout this 34 amino acid tail. It's the 34 amino acid that is minimally required to bind histones. You can see you can't go lower than 34. This is 34 amino acids here showing binding the histone on the top. And on the bottom, you can see where we performed a mutagenesis. And what we found in these mutagenesis studies is that if we make any perturbation to the basic region, you can see that this completely breaks the ability of that 78 amino acids to tether to nucleosomes. Or if we just swap out all of the basic region for alanines, for example, you'll see the same thing. But if we perturb the acidic region, we don't see any perturbation to the ability to bind nucleosomes. So it's really the basic region that's seemingly important for tethering to nucleosomes. Now, if we zoom back out and try to examine these types of mutants in cells, we can see on the left, using ChIP-seq studies, that either the 34 amino acid minimal tail is seemingly performing very similarly to the full fusion on the left-hand side, SS18-SSX. This allows for that very cancer-specific targeting I was describing of synovial sarcoma. But any perturbation to that tail, for example, the two most right columns, the 7 amino acid deletion of the acidic region or perturbing the basic region, prevented entirely that synovial sarcoma specific targeting pattern that we see on chromatin. And this targeting translated into meaningful differences in gene expression as well. You can see only the SS18-SSX full 78 or 34 amino acid fusions produce this very characteristic gain and loss in gene expression, respectively. And you can see that any of these other mutant variants were unable to produce that gene expression. And finally, to examine this in cells, we can see that only the 34 amino acid fusion, for example, was able to rescue proliferation, but any of these mutant variants were unable to do so. So that really begs the importance of that full 34 amino acid tail, and perturbing either the acidic or basic region is important for those properties. We made single residue mutations. For example, this R171A mutation, R169A mutation, excuse me, in that basic region. And you can see that on ChIP-seq on the right-hand side, that this again disrupts, just by a single amino acid, this disrupts the targeting of BAF complexes on chromatin. So to examine where this 78 amino acid tail may be engaging a nucleosome, we teamed up with Tom Muir's lab at Princeton and his very talented postdoc Kadoch, who had developed a method that is essentially based in photo cross-linking using reactive diazurine probes that can be strategically placed throughout the nucleosome to identify sites of unique cross-linking or interaction with any given protein. We chose, of course, the SSX protein, and identified here binding interactions between the E113 residue of H2B and the E56 residue of H2A, whereby in the wild type, we see very strong cross-linking. However, upon making that point mutation in the basic region, the R169A mutation, you can see that that completely goes away. So this allowed us to generate a picture shown on the right of where we think SSX is actually directly binding to the acidic patch. Now, to do these experiments in the reciprocal manner, we also incubated SSX1 protein with nucleosomes that are either wild type in nature, so unmodified mononucleosomes that don't contain any mutations, or 3 different types of nucleosomes that have mutations that disrupt the integrity of the acidic patch. And one can appreciate from this pull-down experiment that with all of these mutations affecting the acidic patch, SSX is no longer able to tether to those nucleosomes. And this was further confirmed using a well-established peptide called alonopeptide that is known to bind the acidic patch. And you can see that this can outcompete at higher concentrations SSX binding. So this is where the story became even a bit more interesting and where actually the study started to converge with simultaneous studies we had going on in the lab focused on intellectual disability syndromes. So Alfredo Valencia in the lab, talented chemical biology graduate student, was simultaneously working on SMARCB1 and noted that in Coffin-Siris syndrome and intellectual disability syndrome, there is a pileup of mutations in this C-terminal coil-coil, putative coil-coil region shown here, that multiple independent individuals with Coffin-Sirius syndrome exhibit this same mutation here, the lysine 364 deletion. And when we examine COSMIC database, you can also see that this was a very frequent site of mutation across a number of human cancers. It's important to note that these are different mutations than the biallelic deletion in malignant rhabdoid tumor. And so what Freddie did is made these mutations and found that uniformly, any mutation in this C-terminal region here was able to completely disrupt the ability of that region to tether to the acidic patch. So in a normal complex, the SMARCB1 C-terminus tethers to the acidic patch. And you can see that any breaking of this interaction resulted in a disappearance of that interaction, and the result of that was a decrease in remodeling activity. If we examine this now structurally, you can see this is this alpha helix here is exactly where many of the mutations lie on this acidic patch interacting site on SMARCB1. And very importantly, we then ask the question, what happens when SSX challenges that SMARCB1 interaction with the acidic patch? So here we incubated nucleosomes with SMARCB1 and competed these with the SSX peptide, and you can see that this can be competed very readily. However, the reverse is not true. If we now incubate nucleosomes with SSX and try to outcompete with the SMARCB1 C-terminus, the SMARCB1 C-terminal peptide is unable to do so, suggesting a tighter, stronger affinity interaction, explaining the results I showed you in the earlier slides of a very uniquely high affinity interaction of this complex with chromatin, and potentially explaining a result that we've had now for many years and had understood until now, whereby what we noticed here back in 2013, when we originally profiled these cell types, we noted that these synovial sarcoma cell lines exhibited a very strong downregulation of BAF47 or SMARCB1 at the protein level, both when we IP the complex or capture the complex, as well as just at the total protein level shown here. This was always a degraded protein. So this competition by SSX for the acidic patch to stabilize the complex on the nucleosome may explain this very selective downregulation at the protein level for SMARCB1. Now, nonetheless, despite the fact that this is now lost SMARCB1 binding to the acidic patch, the SSX interaction with the acidic patch is able to still maintain the ability of BAF complexes to remodel chromatin. You can see that there's a little bit of a decrease here in an in vitro nucleosome remodeling assay, but still these complexes are able to remodel chromatin. And if we examine now what happens in actual cells, you can see that by ATAC-seq, over SS18 and SSX specific sites, we do see DNA accessibility, suggesting the activity of those complexes is still intact. Now, what I told you here is an interaction, a unique interaction that is conferred and gained by having the SSX tail on these complexes, suggesting that it goes to bind the acidic patch and tethers these complexes to nucleosomes in a very, very tight, high affinity manner. But that's insufficient to explain why these complexes go to repressed regions. Why do they selectively target to repressed regions on chromatin? The acidic patch is everywhere on every nucleosome. That doesn't tell us why the complex can go to those specific sites. And we were intrigued to figure that out because if we look at dependency screens, shown here are some of our Project Achilles data, but this was also highlighted by Project Drive from Novartis, you can see that some of the top dependencies for synovial sarcoma tumor types are actually members of the PRC1 complex. So ring 1A, ring 1B members, for example, are one of the highest dependencies shown here. This is unique to synovial sarcoma cell lines and not to a histologic mimic known as SW982 cells, such that these are the top dependencies, ring 1, PCGF3, PCGF5, etc. And we can see this is true in Project Drive data as well, where you can see looking at ring 1B and PCGF3, some of the very top cell lines that were uniquely sensitive to perturbation of those components of PRC1 were indeed synovial sarcoma cell lines. So to try to start exploring that, we first mapped the occupancy of the mark placed by PRC1. This is the mark placed and catalyzed by ring 1A, ring 1B subunits, the H2A monoubiquitination mark at lysine 119. And you can see that this ubiquitination of H2A very strongly overlaps with the sites to which SS18-SSX uniquely targets on chromatin. So these are, again, those repressed sites. You can see this now shown even more clearly when we examine these ChIP-seq plots shown here at the SLIT3 locus, where we see occupancy of the fusion complex, not when we knock the fusion protein down, only in the context of the fusion being present. And we see that H2A ubiquitination is there and ring 1B is there. We can also examine this using imaging and to try to understand which aspect of PRC1 function is really required for the occupancy of these complexes. We examine structurally and use structurally guided mutations of PRC1, where we would make mutations in ring 1A or ring 1B to selectively perturb the ability of that component to place the ubiquitination mark on chromatin. And if you examine the immunofluorescence studies on the right, we can appreciate that in the wild type setting, ring 1B overlaps very nicely with SS18-SSX in cells. They co-target, as I've shown you in many slides before. But if we make these mutations, for example, the R91E mutation or this dual mutation I53A, D56K in ring 1B, you can see that this disappears. So this ability of SSX to localize to these very repressed sites, in this particular cell type, it's actually bar bodies, so an active X chromosome. You can see that that disappears. So this really implicates the function of ring 1B and ring 1A in placing the H2A monoubiquitination mark as the critical nature of the interaction, and suggesting that the H2A ubiquitination is required for this unique targeting that we see with SS18-SSX. So then what part of that tail, that conserved tail that I showed you, may be actually mediating this? So to try to understand if this is true in vitro, we purified SSX again and subjected them to binding assays with either wild type nucleosomes or unmodified nucleosomes, or nucleosomes that actually contain the H2A lysine 119 ubiquitination. And you can appreciate from this Western blot here much increased binding to nucleosomes that contain that modification, as shown here by the histone H3 total blot, suggesting potentially increased affinity to those nucleosomes. I showed you before that the acidic patch binding region, the basic region of the SSX C-terminus, is responsible for tethering the complex to the acidic patch in a unique way. But what about this acidic region? So to go back to that, we knew it's important for activity. We see that now if we incubate either wild type SS18 or SS18 where we've perturbed the acidic region, that last seven amino acid, delta 7 shown there, we can see that this relieves the removal of that acidic region, or perturbation of that acidic region, removes the preference, the preferential binding to H2A ubiquitinated nucleosomes. You can see now that they're equal in comparing the wild type versus the H2A as compared to the wild type SS18. So this unique preference for nucleosomes that are decorated by the PRC1 catalyzed mark, H2A ubiquitination, is mediated by this very conserved acidic C-terminal region. So you have a very interesting interplay between the basic region, which sets up this very unique cancer-specific binding interaction to the acidic patch by SS18-SSX, and this acidic region, which guides the complexes further to want to go to these repressed regions on chromatin. So this leads us to a model in which, in the normal context, you can see that SMARCB1, the complex is arranged structurally in a C shape around the nucleosome. The complex on the core module on the bottom is linked to the nucleosome by the BAF47 C-terminus or SMARCB1 C-terminus. In the presence of SS18-SSX, however, the SSX anchor displaces or overcomes the strength of the interaction to the nucleosome, displacing the ability of F47 to bind the nucleosome and resulting, we think, in its ubiquitinated mediated degradation. And these complexes prefer the orientation of the nucleosome with the H2A lysine 119 ubiquitination. And so with that, I'll stop and thank the members of the lab who did the work. This project was spearheaded by Matthew McBride and Nizar Mashtalir, who partnered very well in this study from both the genomic side and biochemical side. They were aided in analysis by Drew Domino, Martin Filipowski, Evan Winter, and a number of other people listed here. We're very grateful to our collaborators in Tom Muir's lab, Tom Muir and Haidao. And of course, I'm extremely grateful for the funding by Cancer Moonshot, particularly the U54 for the FUSE-ONC2 Consortium, and my many colleagues at NIH that helped make this possible. Thank you.